Hello everyone, it is time for a review before our test on Monday. So we have covered units 1 through 4 so far, and I'm going to briefly go over those 4 units so that you will feel a little more ready for your test. Remember, it'll be more heavily leaned towards 3 and 4, which we have not tested yet. We did have a quiz over unit 3, so I will be going over that in class today. So feel free to go back and watch that video as well. But in the meantime, let us begin with Unit 1. Unit 1 was biomolecules. For biomolecules, we have 4. We have carbohydrates, lipids, proteins, and nucleic acids. Those are our 4 biomolecules. Carbohydrates are those sugars. The monomers are monosaccharide, and the polymer is polysaccharide. All right, remember, monomers are the units, the building blocks. Polymers are a bunch of monomers put together. Now our lipids are the fats, and those are, for the monomers, the glycerol and the fatty acid. And for the polymer, it's those triglycerides. All right, because it has 3 fatty acid chains with the glycerol. Tri means 3. I put the wrong I and Y in the wrong order. Triglycerides, there we go. Now, proteins are what they sound like proteins. Usually they're the meats in the examples. So those are high proteins. The monomer for our proteins are the amino acids. My brain went blank for a second. Amino acids are the monomers, and the polymers are the polypeptides. Lastly, for our nucleic acid, that is either DNA or RNA. This is our genetic information. Monomers are the nucleotides. Remember, these nucleotides are made up of 3 parts. They have a phosphate group. They have a phosphate group, 5 carbon sugar, and our nitrogenous bases. And the polymer are either DNA or RNA. Put the A first, RNA. All right, that's it for biomolecules. The only extra thing is that we have some proteins that are called enzymes. And those enzymes are catalysts that are going to help us make a reaction go faster. So it speeds up a reaction by lowering the activation energy. So there's a certain amount of energy required for every chemical reaction to take place. And what enzymes do is they say, hey, I'm gonna help you out. You don't need that much energy. Here's some energy, here's some stuff. And so it's able to speed up the process because now it no longer needs to have that much energy, right? So that's it for Unit 1. Let's move on to Unit 2. Unit 2, we talked about cell transport. So for cell transport, first we started talking about who controls what goes in and out of the cell. That is your cell membrane. So the cell membrane is made out of a lipid bilayer, lipid bilayer, which means there's two layers of lipids. And remember, we have the heads and the tails. The tails don't like water. All right, so that bilayer controls what goes in and out of the cell because our cell wants to maintain homeostasis. And I can't really see the orange on there. I don't know if you guys can. So the heads like the water, the tails don't like the water. That is going to control what goes in and out of the cell through the spaces between those layers. Now, sometimes we have these proteins along the membrane through which some larger molecules can get by. These are called protein channels or protein pumps, depending on the whether they use energy or not. So we have a few processes that take place in order to transport things in and out of the cell. So we have first, which one did we talk about first? Trying to go by the notebook to make sure we're on the same page. So we talked about the passive transport first, which means it does not require any energy. Passive equals no energy. Okay, and we have three kinds of passive. We have simple diffusion, facilitated diffusion, and osmosis. So simple diffusion is when things go through the tiny spaces between the phospholipids, in the cell membrane. So if they just go through those spaces in the membrane, that is simple diffusion. Now, facilitated diffusion is when they can't fit in between the spaces of the phospholipids, and so they have to go through a protein channel, which is just like a hallway for the molecules to go through. That's a little larger. That's it. It does not do anything else. Lastly, osmosis is simple diffusion. It's the same process, but with water. So osmosis is simple diffusion, but only for water. Okay? Sorry, facilitated diffusion, because water is too large to get through the phospholipid bilayer. It goes through the protein channel. So osmosis is facilitated diffusion, but specifically for water. So if we're moving any other molecule through those protein channels, it's facilitated diffusion. The moment you talk about water, that's osmosis. So if you see a problem that talks about water, immediately that should go to your head, osmosis. Okay? And then we have the active kind of transport, which does require energy. And again, we have three kinds. The first kind is endocytosis. Second kind is exocytosis. And the last one are protein pumps. Okay, so in endocytosis, endo means enter. So we're going to have larger molecules enter the cell. Now for this process, it will require energy because these molecules are too large to move on their own. So energy will be needed there. Now exocytosis is the same thing, large molecules, but now they exit the cell. That's it, okay? Now through this process, endocytosis and exocytosis, we're using vesicles. So either for endo, it creates a vesicle from the cell membrane. And for exocytosis, it uses a vesicle already inside of the cell and moves it out. All right. Now our protein pumps, they are similar to our protein channels, but they require energy. So you have to pay in order to move along. Okay. So our most common one that we use are the sodium-potassium pumps, and those are going to require energy. Now, the energy that we use in active transport is called adenosine triphosphate, or ATP for short. Okay. Now, remember, for cell transport, we are trying to maintain homeostasis. We are trying to maintain balance inside and outside of the cell. So we want there to be a balance, and so normally your molecules would move from an area of high concentration to an area of low concentration, right? And when that happens, that is passive transport. It does not require any energy. Think of it like a slide. It's easy for you to slide down. Gravity's helping you, right? You're going in the direction that the world wants you to go in. However, if you are that stubborn child that wants to go against the grain, you want to go up the slide, then you're going from low to a high concentration. You're going to need energy. In molecules, our energy is ATP. Okay? That's basically it for Unit 2. Oh, tonicity. Lastly, depending on the concentration inside and outside of the cell of water specifically. So remember this osmosis process. That scared me. Okay, so when we say something is hypertonic, that means there's a lot of water inside the cell and not as much water outside of the cell. So this large cell is going to want to shrink and give some of its water to the outside area. So in hypertonic, the water leaves. Now, in hypotonic, it's the opposite. Water's going to enter. For that water to enter, then it means the cell was a little smaller first. And after we put water in the cell, it's going to get a little larger. So it's going to grow. And lastly, we have isotonic, which means there's an equal amount of water inside and out. So it doesn't mean that there's not going to be any motion. It's just going to be going in and out at the same time by the same amount. And that's it. Think of it like a bucket of water, okay? If your bucket of water is full, you're going to want to empty it, right? And use that water in other things. What if you need to move water somewhere else? Then you're going to need to fill up the bucket, right? So that's the hypotonic. You're adding water to the bucket. Now in isotonic, it's a bucket that has water in it, and you're still connecting the hose into the bucket. So the amount of water that's going into the bucket is also going to go out of the bucket because it's full, right? So as the water's coming in, water's spilling over. That's isotonic, okay? Then we move on to Unit 3. This is a terrible eraser. All right, Unit 3, we talk about DNA and RNA. So for DNA and RNA, we had our DNA structure. So our DNA structure is a double helix. So let's remember that. Our double helix shape looks something like this. It's kind of like a twisted ladder because it has steps here connecting to each other, which are the nitrogenous bases, which in turn code everything about you. So if we zoom in to each nucleotide on this DNA, what makes up the actual ladder, the actual double helix, that's what we call the backbone. And the backbone is like the main structure. That is where we have the phosphate group and the sugars. So this backbone, the brace of the DNA, is made up of the phosphate and the sugars. And then the last part of the nucleotides, which is the nitrogenous bases, are the steps inside of the double helix. Okay? Now, these steps in between, we have those hydrogen bonds holding them together, and that is the main structure of DNA. Now remember, the order of the nitrogenous bases, also sometimes called the sequence, that is what's going to tell you your genetic traits, right? That's what's going to give you what genes will show up for you, whether you're going to have blonde hair or blue eyes, or brown hair and green eyes, or black hair and brown eyes, whatever, right? So it's going to determine all of those genetic traits based on the order that these nitrogenous bases are in your DNA strands. Now remember, DNA is double-stranded, which means it has two of these backbones, okay? So this is DNA. Now the difference between DNA and RNA is that RNA is a single-stranded thing. So it's still made up of the same things. This backbone is made out of phosphates and sugars. And then you have one set of steps. So you don't have any hydrogen bonds here in the RNA. You just have the nucleotides all connected into a long chain. But they're not connected to anything else on the other side. They're still in that helical shape, but there's only one side of the ladder. So that's your RNA. That's it. DNA is called DNA because the sugar that it's made out of is called deoxyribose. Whereas in RNA, the sugar is just ribose. Remember, this ose ending is what tells you that it's going to be a sugar. For enzymes, that would be ase. Sugars, ose. Make sure you remember your prefixes and suffixes. Now, we also talked about DNA replication. For DNA to be replicated, we go through a three-step process. Step one, we get the helicase enzyme, go through and unzip your DNA. So it's going to break those hydrogen bonds between the nucleotides and separate them out. That's the first step. We're breaking the bond. Those steps are no longer connected. There's space between them. Step number two, that's going to be the DNA polymerase. So we're going to have one strand here and the other strand backwards, the other strand there. So now that they're separated, the DNA polymerase is going to come in. So step one, step two, in each of the strands, the DNA polymerase is going to match it up with the nucleotide it's supposed to have on the other side. All right, so it's just going to add the missing nucleotides that we took away, and it's going to be doing the same thing on both strands at the same time. So now we're going to have 3 strands of DNA. Okay, sorry, 4 strands of DNA. We had our original 2 and the new 2 that the DNA polymerase is adding in. And then our third and last step, the ligase is going to come in and seal them together. So it's going to add those bonds in between the nucleotides, and that's going to seal 2 different DNA strands. And so now we went from having 1 strand to having 2 full strands. So now what we have is 2 double helixes that are exactly identical. I know my drawing's not identical, but you get the gist. So helicase breaks them open and pushes them apart. The DNA polymerase makes up the other side for each of the 2. The ligase glues them together. And that is it for Unit 3. That's all we talked about. DNA and RNA differences and the steps of DNA replication. All of this should be in your notes in your notebook. Lastly, Unit 4. This is when we talked about cell growth and cell cycle. So for the cell cycle, we have a few steps. Step number 1 is interphase. And step number 2 is mitosis. Step number 3 is cytokinesis. So the interphase is the preparation step. The mitosis is the actual division step. And cytokinesis means cell movement. So it's going to have the 2 new cells separate. All right, so that's your 3 steps. You prepare during the interphase for division. You actually divide during mitosis, and then the cells separate in cytokinesis. Now, interphase and mitosis are both made up of steps. So in the interphase, we have 3 steps. We have the G1 phase, which is the first growth phase. Then we have synthesis, which is when we do the DNA replication. The steps that we just talked about like a couple of minutes ago, where we have the helicase, the DNA polymerase, and the ligase, that process of making from 1 DNA 2 DNAs, that happens during the S phase in interphase. And lastly, after that DNA is replicated, we have the G2 phase, which is where we create the rest of the copies of the organelles and make sure that everything is ready to divide. So now you have 2 of everything. The cell is fully ready to move on into the division. So during mitosis, our first step is prophase. During prophase, we have the nucleus membrane disappear. So we no longer have a nucleus. The DNA will now be floating freely in the cytoplasm. So prophase, we get rid of the nucleus. Then we have metaphase, which is when our chromosomes line up in the center of the cell. Then we have anaphase, which is when those spindle fibers connect to the chromosomes and start pulling them. So in anaphase, we connect to all the chromosomes. We get telophase, and everything moves towards the 2 poles in the cell. So all the DNA material, half of the DNA material goes to one side, the other half goes to the other side. And then we're done with mitosis. So now we basically have 2 cells just in the space of one. So each one has their nucleus, each one has its organelles, and they're far apart from each other. And then cytokinesis comes in and it just breaks the cell. And now you have 2 complete cells. That's it for the cell cycle. We also talked about what happens if the cell cycle goes wrong. So we have these regulators that are going to make sure that everything is going perfectly as we move along through the steps. And those are proteins, specially called cyclins. So these cyclins are going to regulate this process. They're going to make sure all the steps are happening in the right order and that everything is 100% ready. So these are going to have a few checkpoints along the way. You don't have to memorize where the checkpoints are, that's fine. Just know that there are checkpoints both in the interphase and mitosis stages to make sure that everything is flowing correctly. Whenever we have something that does not go correctly, you can have cells that are reproducing too quickly, and some of them may be cancerous cells. And those cancer cells are going to be very difficult to deal with because they are moving very, very fast. They are making a lot of other cells just like them. And so when we get a huge clump of a bunch of cancer cells together, that creates tumor tissue. Déjeme 3 por si acaso. Gracias. So we have a bunch of cells, and that's going to create cancer tissue to grow, and that can develop into tumors. Remember, we have our 2 kinds of tumors. Malignant are bad, malos. The benign are good, buenos. They start with the same letter, so remember that. Bad is malignant. Good is benign. Maleficent is bad. Or she's not really in the new movies, but you know what I mean. Mal, bad. Benign, good. So those are kinds of tumors which can be caused if the cell division is not properly regulated. Now that we know that, we also know that the P53 gene is a gene that can help us detect tumors early. They are able to notify when something is going wrong. So it's a very important gene to know about. Now, what else? Cell differentiation, last topic that we talked about. To differentiate, each cell in your body starts off the same. Those are called stem cells, right? As soon as the cells duplicate, they finish the cell cycle, you have 2 identical cells. So depending on different things, both internally and externally from the cell, it's what's going to determine what that cell is going to work as. Is it going to be a skin cell? Is it going to be an eye cell? Is it going to be a muscle cell, bone cell, right? It depends on what we need. So inside of the cell, what's going to tell the cell what to do is your DNA. So you have the whole DNA thing inside each cell, but each cell does not represent all of the DNA. Only a portion of the DNA lights up, right? It activates. It is expressed. So we call it gene expression, the fact that only a certain gene is activated in each cell. So the gene that is activated for that specific cell is going to tell it, hey, this is what I need you to do. So that's the internal factors. External factors can also affect the the gene expression in the cell. So depending, for a same species, for example, some species need to have thicker fur depending on the climate. So that gene will be expressed. It depends on the inside and it depends on the outside. So environmental factors play a role, and then which gene is expressed inside the DNA of that cell also plays a role. Lastly, the location of the cell in the body can also be an indicator of what it's going to be. So based on the location, you can determine whether it's going to be a skin cell, or is it going to be a toenail cell, right? Depends on where it is. If it's in your cheek, then you're going to need skin cell or muscle cell, right? If it's in your toe, you might need a toenail cell. Depending on location, it can also help guide the cell to its correct future path. So that's it for all 4 units. I know this was probably pretty long, so I'm going to let you guys go now. If you still have any questions, there are plenty more videos going in depth into each topic, both here on the playlist and in my team's channels. So I will be posting this very soon, so hopefully you get weekend to go through it, go through your notebook. It's going to have all those important notes that you're going to need to study. And that's it for me. I will see you guys on Monday. Take care. Bye-bye. Where's the goodbye?